Welcome, my dear students. Today we explore another exciting and fundamental concept on biochemistry, the electrochemical properties of amino acids. Have you ever wondered how amino acids behave in different pH conditions, or why the same molecule can carry a positive charge, a negative charge, or no charge at all? Amino acids are unique because they act as both acids and bases, and this dual nature influences protein structure, enzyme activity, and countless cellular processes. In this class, we will uncover how amino acids ionize, how their charge changes with pH, and why concepts like pKa, Zwitterion, and isoelectric point are so important in biology. Electrochemical properties of amino acid. What do you mean by electrochemical properties of amino acid? The electrochemical properties of amino acids refer to how they behave electrically and chemically in different environments, especially in water. These properties arise mainly because each amino acid contains an amino group, that is NH2, or bond NH3 plus, that is basic in nature, and a carboxyl group bond COOH or bond COO minus, an acidic group. A variable side chain or R group gives individual amino acids unique electrical behavior. These make amino acid amphoteric, meaning they can act as both acids and bases. So what is Zwitterion? How Zwitterion formation occur? In aqueous solution like inside our body, amino acids do not exist as neutral molecule. They exist as Zwitterions, molecules that have both positive and negative charges. The amino group becomes NH3 plus positive, the carboxyl group becomes C double minus negative. So the net charge will be 0. This gives amino acid special electrical properties. This is the amino acid which have C. This is the basic group of the amino acid, and this is the acidic group of the amino acid. There is an equilibrium between this amino acid and its deuteronic state. When the amino acids in aqueous solution, it acts as NH3 plus and C double minus. Amphoteric nature, amino acids can donate H plus when it acts as acid, and accept H minus, accept H plus when it acts as bases. So C double H is equal to C double minus plus H plus, that is acidic, and NH2 plus H plus is equal to NH3 plus, that is basic. So this dual behavior is crucial in buffering, protein folding, and pH regulation. Electric point, isoelectric point, or pi. The isoelectric point is the pH at which an amino acid carries no net charge. At pi, amino acid is in deuterionic form. It is less soluble in water. It does not move in an electric field, so it is important in electrophoresis. Different amino acids have different pi values depending on their side chain charges. Ionization of R groups. Some amino acids have ionizable side chains. Acidic amino acids are aspartic acid and glutamic acid. They carry negative charge. Basic amino acid, lysine, arginine, histidine, they carry positive charge. Others may be either polar or non-polar. These ionizations control the protein structure, enzyme active sites, binding interactions, and catalytic activity. Electrical conductivity. Because amino acids exist as charged species in solution, they can conduct electricity. This property is utilized in electrophoresis and chromatography, protein separation techniques. Amino acids move toward the electrode opposite their charge. Positive amino acids move toward cathode, and negative amino acids move toward anode. Buffering action. Amino acids resist sudden changes in pH due to their ionizable groups. So each amino acid has two buffering regions, one due to COOH and one due to NH3 plus. Amino acids with ionizable R groups have three buffering regions. Redox properties, which is found in special cases. Some amino acids are there which can participate in oxidation, reduction, redox reactions, such as example cysteine, which forms disulfide bond, SS bond, and it is important for protein folding and stability. Amino acids can act as acids and bases. When an amino acid is dissolved in water, it does not stay as a simple neutral molecule. Instead, it becomes a special type of ion called a Zwitterion, from a German word meaning hybrid ion. A Zwitterion has two opposite charges at the same time. The amino group bond NH3 carries a positive charge, and the bond carboxyl group bond COO minus carries a negative charge. Because it has both positive and negative charge, the amino acid can behave in two ways. That is, it can act as an acid by donating a proton or H plus, and can act as a base by accepting a proton. Substances that can act as both acids and bases are called amphoteric or ampholytes. Ampholytes means amphoteric electrolytes. So a simple amino acid like alanine has one carboxyl group and one amino group. These two groups can release protons. So a fully protonated amino acid behave like a diprotic acid, and that give two H plus ions. Amino acids have characteristic titration curves. What do you mean by titration curve? A titration curve of an amino acid is a graph that shows the pH changes when you add acid or base to the amino acid solution. When we try titrate an amino acid like glycine, we slowly add or remove protons or H plus ions. I know glycine has two groups that can lose protons, such as carboxyl group bond COOH, amino group bond NH3. So because of this, glycine behaves like a diprotic acid, and its transition curve has two steps. At very low pH, which is strongly acidic, at low pH glycine is fully protonated, NH3 CH2 COOH, both groups hold onto their protons. First stage of titration, C bond COOH loses a proton. As we add base, the carboxyl group loses its proton first. At the midpoint of this first stage, half of the glycine molecules are COOH, half are COO minus. So for pH, pKa for bond COOH group, for glycine, pKa1 is 2.34. This means at pH 2.34, the carboxyl group is half ionized. Between the two stages, around pH 5.97, something important happens at pH 5.97. The first proton removal is complete, and the second proton removal has just started. Glycine exists mainly as a Zwitterion, that is NH3 bond CH2 bond COO minus. So the pH 5.97 is the isoelectric point Pi, and the pH where glycine has no net charge. Second stage of titration, there is bond NH3 plus losses a proton. As we continue adding base, the amino group loses its proton. At the midpoint of this second stage, equal amounts of NH3 plus and NH2 forms exist. pH is equal to pKa of bond NH3 plus group. So for glycine it is pKa2 is 9.6. This is when the bond NH3 group is half ionized. Completion of titration around pH 12. At very high pH, titration is complete. Glycine exists mostly in its fully deprotonated form, that is NH2 bond CH2 and COO minus. This is a titration of an amino acid. From the titration curve of glycine, we can learn two main things. Number one is pKa values and why they are different. pKa values of glycine is pKa1 is 2.34 for the carboxyl group, and pKa2 is 9.6 for the amino group. These values show how easily each group loses a proton. Why is the bond COOH group of glycine more acidic than usual? Normally, a carboxyl group, like in acetic acid, has a pKa 4.76. In glycine, pKa is 2.34, which is much lower and it is more acidic. Reason: the nearby positively charged bond NH3 group repels the proton on bond COOH, making it leave more easily. Why is the bond NH3 group positively charged, more acidic than a neutral amine, normal amine. A normal amine like methylamine has a higher pKa. So glycine's bond NH3 has pKa that is 9.6, which is lower than usual. Reason: the electronegative oxygen atoms in the bond COO minus group pull electrons toward themselves. This makes the bond NH3 group more likely to lose its proton. Key idea: the pKa of any group changes depending on its chemical environment. Enzymes also use this phenomenon to help reactions occur more efficiently. Second one is the buffering regions of glycine. Glycine has two buffering zones, one near each pKa. Buffering zone 1, that is around pH 2.34 for the carboxyl group, and buffer zone 2, it's around 9.6 for NH3 plus group. A buffering zone means the pH changes slowly when acid or bases is added. Important note: glycine does not buffer well at pH 7.4, the pH of blood or intercellular fluid. Using Henderson-Hasselbalch equation, within each buffering range, we can use the Henderson-Hasselbalch equation to calculate the ratio of proton donor form and proton acceptor form needed to make a buffer at a specific pH. So pH is equal to pKa plus log A minus by H plus. So the titration curve of an amino acid helps us understand how its electric charge changes with pH. So isoelectric point where pH net charge is 0. For glycine at pH 5.97, it exists mainly as Zwitterion, NH3 plus CH2 and COO minus. So this has no net charge, and this special pH is called the isoelectric point. Because glycine has no ionizable side chain, Pi is equal to pKa1 plus pKa2 by 2, that is 2.34 plus 9.6 divided by 2 is equal to 5.97. Change charge of glycine at different pH values. pH when less than Pi, that is below 5.7, so glycine is positively charged and moves toward the cathode and it is negative electrode, and when pH is greater than Pi, that is above 5.97, so glycine is negatively charged, moves toward the anode, that is which is positive electrode. The further the pH is from Pi, the stronger the charge. Examples: at pH 1, glycine is almost fully protonated, that is NH3 plus bond CH2 bond COOH, where net charge is plus 1, and at pH 2.34, equal amounts of COOH and COO minus forms exist, so net charge is plus 0.5. So key idea: using the titration curve, we can predict whether an amino acid is positive, negative or neutral at any pH, how it will move in an electric field. That is important in case of electrophoresis. So amino acids differ in their acid-base properties. Amino acids behave differently in acid-base reactions depending on whether their R group can ionize. Amino acids without ionizable group, examples glycine, alanine, valine, leucine, etc. They have only two ionizing groups, bond COOH and bond NH3 plus. Their titration curves look like glycine's curve. So their pKa ranges are similar. pKa1, that is for bond COOH, is 1.8 to 2.4, and pKa2, that is bond NH3 plus, the range varies from 8.8 to 11. So these amino acids have two stages in their titration curve and two pKa values. So amino acids with ionizable R group, examples glutamate, aspartate, lysine, histidine, arginine, tyrosine, cysteine. So they have three ionizable groups, bond COOH, bond NH3 plus, and the ionizable side chain or R group. So they have three pKa values and three stages in the titration curve. Examples: glutamate, when it has bond uh, two bond COOH groups, has a low Pi, is 3.22, because extra COOH make it more acidic. Histidine, two groups become positive when protonated and has a high pi 7.59 because the R group or imidazole can carry a positive charge. Buffering near physiological pH. We know most amino acids do not buffer well at pH 7. So histidine is the only amino acid with an R group where pKa is close to neutral pH. This makes the histidine an important buffer in biological systems. So to sum up, the electrochemical properties of amino acids reveal how these tiny molecules respond to change in pH and how their charges shift in different environments. Understanding concepts like pKa, buffering zone, and isoelectric point help us predict amino acid behavior inside cells, in proteins, and even during laboratory techniques like electrophoresis. These properties form the chemical foundation of protein structure and biological function. Mastering them give you the key to understanding deeper biochemical processes essential for undergraduate, postgraduate, and NET life science studies. Thank you.